Hi, my name is Jordan Clark, and today is June 9th of 2020, and today I'm going to be talking about the hormonal control of implantation and early embryonic development. After fertilization in the fallopian tube, the zygote takes about 4 to 5 days to find its way to the uterus. Under the influence of progesterone, the smooth muscle of the tube relaxes and transport proceeds slowly. By the time the developing embryo reaches the uterus, it is a hollow ball of about 100 cells called a blastocyst. Some of the outer layer of the blastocyst becomes the chorion that will enclose the embryo and form the placenta. The inner cell mass will develop into the embryo and 3 other membranes. The first being the amnion, which secretes the amniotic fluid that the embryo floats in. The allantois will become the umbilical cord that links the embryo to the mother, and the yolk sac, which degenerates early in development. Implantation takes place about 7 days after fertilization. The blastocyst secretes enzymes that allow it to invade the endometrium. After it is implanted, the endometrial cells grow around the blastocyst until it is engulfed. As the blastocyst continues dividing and becomes an embryo, cells that will become the placenta form finger-like chorionic villi that penetrate into the endometrium. Enzymes from the villi break down the walls of the maternal blood vessels until the villi are surrounded by pools of maternal blood. Nutrients, gases, and waste exchange across the membrane, mostly by simple diffusion. The tremendous flow to the placenta is one reason abnormal separation of the placenta from the uterine wall is a medical emergency. The placenta secretes several hormones that prevent menstruation during pregnancy, such as human chorionic gonadotropin, human chorionic somatomamotropin, estrogen, and progesterone. The corpus luteum remains active during early pregnancy because of HCG. Under its influence, it keeps producing progesterone to keep the endometrium intact. By week 7 or so of development, the placenta takes over progesterone production and the corpus luteum degenerates. Another function of HCG is stimulating testosterone production in the testes of the male fetus. It is also the chemical detected by pregnancy tests. Human chorionic somatomamotropin, or HCS, is also produced by the placenta. Structurally, it is related to prolactin and growth hormone. It alters the mother's glucose and fatty acid metabolism to support fetal growth. Estrogen and progesterone are produced continuously throughout pregnancy. With high levels of these steroid hormones, feedback suppression of the pituitary gland continues during pregnancy, preventing more follicles from developing. Estrogen contributes to the development of milk-secreting ducts in the breast. And progesterone is essential for maintaining the endometrium and suppressing uterine contractions until it's time for labor. The placenta makes a variety of other hormones like inhibin and prorenin, but most of their functions are still unclear. Parturition, or the process of birth, is when pregnancy ceases. In many non-human mammals, a decrease in estrogen and progesterone marks the beginning. A decrease in progesterone is logical, as progesterone inhibits uterine contractions, but in humans, levels of these hormones don't decrease until labor is well underway. It is still widely unknown what triggers the start of labor in humans. The peptide hormone, oxytocin, that causes uterine contractions is thought to contribute some. Synthetic oxytocin is often given to mothers to induce labor, but it's not always effective. Another possibility is that the fetus somehow signals the mother's body that it has completed development. One theory is that corticotropin-releasing hormone, or CRH, secreted by the placenta, stimulates labor initiation. In the weeks prior to delivery, maternal CRH blood levels increase rapidly. And also, women with elevated CRH levels as early as 15 weeks are much more likely to go into premature labor.